Okay, so here we are for, I think it's going to be could be the year's last research proposal. And it's similar, and he's going to, he's been with us then, well, about 7 months or something since you started.
Yep.
Okay. So he's going to present his work, and then we'll have a little interview with him later on and grill him, which most of you don't have time to do. So, yeah, let's hear it. Thank you very much.
Okay. Good afternoon, everyone. My name is Tim Lim. I'm a PhD student working under Dave Anderson. Today, my research topic is about the chimeric bits and the L1 elements and their impact on the host genome. Host, also known as the Equus colobus, is one of the earliest domesticated animals according to the human historical record. The host family, they are the young evolved species because they diverged into the 8 or 9 species just around 3 million years ago. The host family, they also share one interesting characteristic, where they have exhibit the diverged karyotype and the centromere repositioning, but they are still able to interbreed with each other. For example, like the male is born through the interbreeding of the horse and the donkey. Therefore, the horse is an excellent model for the centromere repositioning and the centromere evolution study. Besides that, the horse is also a good model for the human medical research related to the infertility, inflammatory disease, and the muscle disorder. So most of my PhD research will be focused on the host genome. So today I will give a brief introduction about the thrombosomal elements. Then I will talk about the impact of the thrombosomes, especially the L1 elements, to the genome structural variation. After that, I will introduce the chimeric bits and discuss the current questions related to it. And finally, I will present my PhD research proposal. Thrombosomal elements, they are also known as the mobile elements and jumping genes because they have the potential to move around to different genomic locations, either inside a nucleus or from cell to cell. They were first discovered by the Max Hinson back in 1951, and at that time, the thrombosomes were known as the genome parasites because the scientists believed that these thrombosomes, they try to use their full potential to replicate faster than the host genome in order to survive. However, recent studies suggest that these thrombosomes, they are not only able to create a new gene function, but they shape up the eukaryotic genome step by step through the evolution. And then, basically, there are two major classes of the thrombosomal elements: the class 1 retrothrombosomes and the class 2 thrombosomes. For the class 2 thrombosomes, they are also known as the DNA thrombosomes because they transpose over the genome in the DNA intermediate form. So during the thrombosis event, these DNA thrombosomes, they will excite from the original genome location and appear in the double-stranded DNA intermediate form. And this intermediate form, they will just integrate to somewhere else in the genome through the cut and paste mechanism. However, for these DNA thrombosomes, they are only a small fraction inside the human or other mammalian genome due to their limited movement and non-duplicability. For the class 1 retrothrombosomes, it can divide into two major types. The first type is the LTR retrothrombosomes and the endogenous retroviruses. And the second type is the non-LTR retrothrombosomes, which can further subdivide into the autonomous L1 element and non-autonomous signs. Although the LTR and the non-LTR retrothrombosomes, they have different structure and mobility, but they share one same characteristic, where they are replicated by their RNA copies through the copy and paste mechanism. Which means that the original retrothrombosomes, they will remain in the same genomic location, but their new duplicated copy will be just integrated somewhere else. For this copy and paste mechanism, they are much more efficient than the DNA thrombosomes cut and paste mechanism. Therefore, for the class 1 retrothrombosomes, they comprise around 50% of the mammalian genome. For the LTR retrothrombosomes and the endogenous retroviruses, basically they have the long terminal repeats in the 5' and the 3' region. And they also contain the multiple regulatory elements for the retrotransposition purposes. For example, like the GAP and the POL gene. And the envelope gene is registered specific for the endogenous retroviruses only. So during the retrotransposition event, the LTR retrothrombosomes, they will first transcribe into the RNA copy. And this RNA copy, they will reverse transcribe back to the cDNA. And this cDNA, they will form a pre-integration complex, and after that, they will just integrate to the host genome. For these LTR retrothrombosomes, basically they are quite specific because they only integrate into the specific part of the genome, which is not like the normal non-LTR retrothrombosomes. Because the non-LTR retrothrombosomes, they will be just integrate randomly in the host genome, which I will talk about it in the next 2 slides. For the non-LTR retrothrombosomes, as the name suggests, they don't have the long terminal repeats in the 5' and the 3' sites. And I will talk about the autonomous L1 element first. For an active copy of the L1 element, it has a 5' UTR region, which contains a possible RNA polymerase promoter, the OFF1 and the OFF2 gene for the retrothrombosis purposes. So during the retrothrombosis event, first the L1 DNA they will transcribe into the RNA copy form and export to the cytoplasm. In the cytoplasm, the OFF1 and the OFF2 encoded proteins they were translated into the OFF1P and the OFF2P. The L1 RNA, OFF1P and the OFF2P they will form a L1 ribonucleoprotein complex and invert back to the nucleus. And for this complex, they will just render in the random part of the host genome. And then the L1 RNA, they will start to be reverse transcribing back to the DNA form and integrate to the genome through the cis preference mechanisms. For the non-autonomous sites, basically they're also known as the parasites of the parasite, which I will talk about it later on. So for this non-autonomous site, it has a non-coding RNA in the 5' part, which acts as an internal promoter, and any non-coding DNA sequences in the 3' region. So during the retrothrombosis event, this non-autonomous site DNA will first transcribe into the RNA copy, and this RNA copy, they will hijack the OFF1 and the OFF2 P proteins from the L1 element in order to help them to reverse transcribe back to DNA and integrate back to the genome through the trans mechanism. This is why they're known as the parasites of the parasite, because they really need to rely on the L1 element protein in order to have a successful reverse retrothrombosis event. So, in the beginning, I already mentioned the importance of the thrombosomes to the eukaryotic genome. However, the real impact of the thrombosomes, especially the L1 elements, they are not only able to shape up the host genome structure, but they create the unique individual genome structural variations through their L1 copy number variation activity, which I will discuss about it in the next few slides. Structural variations, in a single definition, is the rearrangement or shuffling of the DNA sequences, which will change the orientation of the host genome structure. Basically, there are three different types of the structural variations: SNPs, single nucleotide polymorphism, indels, swap insertion and the deletions, and CNVs, the copy number variations, which I am interested in. The definition of the copy number variation is the presence or absence of a singular copy of the gene inside the host genome. However, they are not easy to study or identify compared with the SNPs or the indels. For example, like the tandem repeats and the thrombosomes. Although they share the common features of the copy number variations, but their characteristics are quite distinct with each other. In the past, the copy number variation study in the human genome is extremely hard due to the technology limitations. At that time, different labs, they are using a different experimental method, different data processing algorithm with different levels of the standards. Therefore, their CNV study often contain a high false positive and a false negative rates of the results. And it is even worse when there's only a limited human genome sample available at that time. However, recent studies done by few independent groups in last few months, they suggest that the L1 copy number variations, they have a major impact to the human genome with a very strong evidences. So these are the experiments they have done. So in their study, they have identified a different unique insertions of the L1 elements in the different human genome samples through different experimental methods, such as the microarray, L1 sequencing assay, computational study with the L1 cell culture, and then the DNA sequencing method with a really high quality of the control. In their study, they estimate that in every 95 to 270 of the new births, there is at least one new birth contain a new L1 insertion. Therefore, they conclude that these L1 copy number variations, they not only create the unique individual genome for every one of us, but it may also lead to the species speciation as well. For example, like the divergence of the chimpanzee and the human, possibly due to these L1 copy number variation activity. In the beginning, I also mentioned the importance of the host as the research model to the community. Therefore, in order to understand the host genome in depth, the host genome sequencing project was started from 1995 and finished last year. In the host genome, the distribution of the transposable elements are quite similar with other mammalian genomes. However, there's still around 11% of the transposons that still remain unclassified. And because they are unable to annotate by the repeat masker. So when we have a detailed look on these unclassified transposons, they look quite funny because they doesn't belong to any single classes of the transposable elements, but they are known as the chimeric repeats, which is still remain a mystery at the moment, which I will talk about it in the next few slides. Chimeric repeats. The simplest definition for it is the hybrid or fusion of few different non-transposable elements. For this type of chimeric repeats, they are unable to identify by the current repeat master software because they don't share any homologs with the any well-studied transposable element. So right now, there are two different types of the chimeric repeats classification proposed by the other scientists, which I will talk about it later on. Before I move on, I would like to point out that in my PhD research, I will only focus on those bipartite and the tripartite chimeric repeats because the formation of the chimeric repeats with more than 3 different transposable elements, they are way too complicated and unable to study at the moment. So I will ignore them for now. The first type of the chimeric repeats classification is quite straightforward. It's just the fusion of 2 or more than 2 different types of the non-transposable elements. The best example is the SPA element that is covered in the human genome only. For this SPA element, as you can see, they contain the alphabet sequences, variable number tandem repeat region, and the sign sequences. And for the second type of the classification is much more specific, where there must be a non-coding RNA in the 5' part and any non-transposable elements in the 3' region. For this type of the chimeric repeats, basically their copy numbers are quite lower than the first type due to their specificity. So, what's so special about these chimeric repeats compared with the normal transposable elements? First, for the human chimeric repeats, they're expressed in the specific tissue cell, and it is grown through the in vitro study. Therefore, it suggests that these chimeric repeats they may play an important functional role in the human body, or they still have the potential to be retrotransposed. The other interesting fact about these chimeric repeats is they are the linear